Hello and welcome everybody to my channel Omni Bioentrance, and today we will be seeing Kerala SET Biotechnology question paper conducted in the year July 22. I have discussed biotechnology question paper of Kerala SET exam, state eligibility test. This can be seen as a model question paper for other SET exams also conducted by different states. So coming to the first question. Actin filament is composed of troponin, tropomyosin, actin, all of the above. Answer is option D. Different serine proteases differ in. So they differ in their substrate specificity. Such as if you take the serine proteases, chymotrypsin and trypsin, you know that chymotrypsin cleaves on the C terminal of phenylalanine, tryptophan and tyrosine, whereas trypsin cleaves on the C terminal of lysine and arginine residues. So they differ in their substrate specificity. Purity of the isolated protein can be checked by. So it can be done by UV visible spectroscopy using Bradford's or other activity assays. Then electrophoresis, it provides a picture on the size of your target protein and also whether other protein based impurities are present in it. So that is what electrophoresis does. Then hydrophobic interaction chromatography, it separates protein on the basis of their hydrophobicity. So purity of isolated protein can be checked by electrophoresis and UV visible spectroscopy. Yes. Electrophoresis and immunodiffusion. No. Immunodiffusion cannot be used. UV visible spectroscopy and hydrophobic interaction chromatography. Answer is option D, that is both A and C are correct. Then fructose is the energy source for sperms and is produced by the action of. So fructose, it is secreted from the seminal vesicles and the accessory sex glands. It is the carbohydrate source in the seminal plasma and is essential for sperm motility. So it is produced by the action of aldose reductase and sorbitol dehydrogenase. Answer is option C. The next one. The ion needed for the storage and action of insulin. It is Zn2 positive ion. Then Ozazone test can be used for distinguishing. So Ozazone test, it is used to detect reducing sugars. It differentiates between reducing and non-reducing sugars. Then it is also used to distinguish between reducing sugars between each other. So first is sucrose and starch. Sucrose is non-reducing and starch is also a non-reducing sugar. So Ozazone test cannot be used for distinguishing them. Then starch and glycogen. Glycogen is a polysaccharide. So it is not used to detect Ozazone test. Then fructose and lactose. Fructose will give needle shape Ozazone and lactose will give cotton ball or powdery puff shaped Ozazone. So fructose and lactose can be distinguished by Ozazone test. Why cannot glucose and fructose? Because during Ozazone formation, the reaction, it occurs mainly at the C1 and C2 of carbon atoms. And glucose and fructose, they differ from each other only in the arrangement of atoms at C1 and C2. Therefore, they will give the same Ozazone. So they cannot be distinguished by Ozazone test. So the answer here is fructose and lactose. The next one. When the rotor of ATP synthase rotates 360 degrees, 3H positive ions are shuttled back to mitochondrial matrix. That itself is correct. So option A is correct. A characteristic of animalia: absence of cell wall, presence of chloroplast, athetated nucleus, presence of mesosomes. So absence of cell wall, that is the characteristic of animalia. Cell wall is not present in kingdom Animalia. The next one. The most common cell wall component of fungi, it is chitin. Chitin is the most common component of fungal cell wall. Then next one, a virus with single-stranded DNA genome. So lambda phage, it is linear DNA and is double-stranded. TMB is single-stranded positive sense RNA. F1 phage is circular single-stranded DNA, and CAMV is double-stranded DNA. So they have asked for single-stranded DNA genome, so it is F1 phage. Then myotonia congenita is caused by. So myotonia congenita, it is a neuromuscular channelopathy that affects the disease that affects the skeletal muscles, and it's characterized by delayed relaxation of the muscles, that is called as myotonia, and also rigidity. Then this disease appears to be linked to CLCN1 gene that is required for the normal functioning of the chloride channels in skeletal muscle cell membranes. And if there is a mutation in the CLCN1 gene, or there is reduction in the number, if there is a mutation in CLCN1 gene, there will be a reduction in the number of the chloride channels. So myotonia congenita is caused by mutations affecting chloride channel. That is why it is called as neuromuscular channelopathy. Channelopathy is a disease that affects the channels. So here it is chloride channel that is affected. Then P elements in Drosophila cause PM hybrid dysgenesis used as vector for Drosophila. These are the two statements that they have given. 1 and 2 are correct, and 1 is the correct explanation of 2. So you have to see from the options which is correct and which is giving the correct explanation. So before that, what are P elements? They are transposable elements and they are causative for hybrid dysgenesis in Drosophila. What is hybrid dysgenesis? Hybrid dysgenesis is resulting from a cross between males which have P elements, that is P strain, and females that lack the P elements, that is M strain. So P elements is the cause of PM hybrid dysgenesis, that is correct. Used as vectors for Drosophila, that is also correct. So 1 and 2 are correct, and 1 is not the correct explanation of 2. Answer is option B. Then next one, the deamination of 5-methyl cytosine results in. So 5-methyl cytosine, so this is your normal CG DNA strands having cytosine and guanine, normal DNA strand. Once it replicates, if C is converted to 5-methyl cytosine, is existing as 5-methyl cytosine, it will pair normally with G. But if there is a deamination, this deamination of 5-methyl cytosine results in it becoming a thymine. So if there is a deamination, it will behave like thymine and it will pair with adenine. Whereas this particular strand, it will be normal. So here it will pair with adenine. So in the next division, there will be 5-methyl cytosine and A. So 5-methyl cytosine, it is deaminated, it will pair with A, and A will pair with T. So a CG mutation has been converted to an AT mutation. So in place of guanine, adenine is coming, so it is a GA transition. Answer is option B. Then RB1 mutation causes cancer by. So RB1 gene, it is the first tumor suppressor gene and whose inactivation causes retinoblastoma. So cells with the 2 altered copies of RB1 gene produce no functional PRB, and it is unable to regulate the cell division. So as a result, retinal cells lacking functional PRB will divide uncontrollably, resulting in cancer. So RB1 mutation causes cancer by loss of function. Once its function is lost, It will result in cancer. Then uncompetitive inhibitor. Uncompetitive inhibitor decreases both Km and Vmax value. These are usual questions for most of the exams. Uncompetitive, competitive, non-competitive inhibitor. What effect they have on the Km and Vmax value? It is better if you study all those things. They will be usually asked for most of the entrance examinations. Then rate zonal centrifugation separate particles that differ in. So that differ in size but not density. Answer is option A. The commonly used reference electrode in pH meter is dash electrode. It is silver chloride. Answer is option C. Then relationship between absorbance and transmittance is option C. A is equal to log to the base 10 1 by T. Then next one. In HPLC, peak area refers to. So this is the graph of an HPLC output. So this is the retention time and this is the response. So this is the peak width. This distance, that is the peak width. This is the baseline. This is the peak width. This is the peak height. These are the various data points and this is the peak area. So they have asked peak area refers to the concentration of the compound. Answer is option A. The next one. Which of the following compound releases more energy upon hydrolysis? So it is creatinine phosphate that releases more energy upon hydrolysis. Answer is option C, not ATP. Then Lin-Weaver-Burke plot can be used for analysis of bisubstrate reaction, analysis of enzyme inhibition, kinetic analysis. It can be used for all these three analyses. So answer here is option D, that is all of the above. Then Philadelphia chromosome causes chronic myelogenous leukemia. Statement 1. Philadelphia chromosome has ABL-BCR fusion. Statement 2. Philadelphia chromosome increases tyrosine kinase activity in cells. So, what is this Philadelphia chromosome? The ABL gene from chromosome 9 joins to BCR gene on chromosome 22 to form BCR ABL fusion gene. This change in chromosome 22 result in what is called as Philadelphia chromosome. And this BCR ABL fusion, it is found in patients with chronic myelogenous leukemia. And the product of this fusion, that is BCR ABL gene, is a constitutively active protein tyrosine kinase that promotes cellular proliferation and suppresses apoptosis. So answer here is both 1 and 2 are correct and 1 is the correct explanation of 2. Then next one. Triticum aestivum is the bread wheat. Statement 1. Bread wheat is fertile hexaploid. That is correct. Bread wheat is an amphidiploid. That is also correct. So both 1 and 2 are correct. Then next one. The perfect population size of a trihybrid cross. So it is trihybrid. So how many gametes will be produced? 2 raised to n. There are 3 heterozygotes. So 2 raised to n means 2 raised to 3. So it will be 8 gametes. So when you draw the Punnett square, you will get 8 into 8, that is 64. So what will be the population size of a trihybrid cross? It will be 64. Then which among the following is not a natural gene transfer mechanism in bacteria? So it is transduction, it's a natural mechanism. Transfection is not in bacteria. It is the process of deliberately introducing naked or purified DNA into eukaryotic cells. That is called as transfection. Then conjugation and transformation, they are natural gene transfer mechanisms in bacteria. Then the Drosophila genes which show crisscross inheritance. So what is crisscross inheritance? That is a character from the father goes to the daughter and then from the daughter to the grandson in the next generation. That is called as crisscross inheritance, as you see in many X-linked diseases. So in Drosophila it is red and white type alleles. Answer is option A. Next question. The dash affect the HW equilibrium. HW is Hardy-Weinberg equilibrium. So what does Hardy-Weinberg law states? It states that genotype and allele frequencies, they will remain constant from generation to generation if certain conditions are followed. So what are the conditions? The population should be large, it should mate randomly, and there shouldn't be any natural selection, mutation, immigration, etc. in the population. So that is what Hardy-Weinberg equilibrium states. So dash affects Hardy-Weinberg equilibrium. Large population, no. Cross fertilization, no. Diploidy, no. Natural selection, yes. Natural selection will affect Hardy-Weinberg equilibrium. Then next question. And Hardy-Weinberg equilibrium is a sure shot question for most of the exams. One question you can expect from Hardy-Weinberg law, either in the form of a problem or as a theory based question. I have done other Kerala SET Biotechnology exams from 2019. I have answered all those from 2019 to 2022. I have answered all the previous year questions. You can refer to my channel for seeing and learning those question papers. Then, in a Hardy-Weinberg population, the individual with AA genotype have a frequency of 0.49. Find out the frequency of heterozygotes in the population if AA, capital A, and small a are the only genotypes in the population. So they have given us the frequency of AA genotype. So it is P square, so it is 0.49, so P is 0.7. We know that P plus Q is equal to 1 as per Hardy-Weinberg equilibrium. Q is equal to 1 minus P, that will be 0.3. Then they have asked for frequency of heterozygotes in the population. So it is 2PQ. So 2 into 0.7 into 0.3, that is 0.42. So the answer is option D. Then next one. Disinfectant inactivate or destroy microorganisms. So disinfectants inactivate or destroy microorganism on inert surfaces. Answer is option C. Then passive agglutination employs carrier particles that are coated with. So passive agglutination employs carrier particles that are coated with soluble antigens. And these carrier particles, it include erythrocytes or lactose spheres. And when specific antibodies are added to these the antigens, there will be agglutination reaction taking place. So the answer here is option A, that is soluble antigen. Then CD is a cluster of differentiation as well as designation. Both you can say. So answer is option C, that is both A and B. Then opsonins. Opsonin enhance phagocytosis. That is correct. So option A itself is correct. Then cholesterol is not present in. So cholesterol is not present in bacteria. Whereas influenza virus cell membrane, endoplasmic reticulum membrane, cholesterol is present, whereas it is not present in bacteria. Then reacting with Benedict's reagent, glucose is converted to. So glucose is having an aldehyde group, it is an aldose, and an aldose reacts with the Benedict's reagent forming carboxylate anion, or here it is gluconic acid. So answer here is option B, that is gluconic acid. And what is Benedict's reagent? It is a complex mixture of sodium citrate, sodium carbonate and pentahydrate of copper sulfate 2. Next one. An example of reversible kinase reaction is the reaction catalyzed by. So glucokinase, it occurs in liver and pancreas, and hexokinase, both of them it facilitate phosphorylation of glucose to glucose 6-phosphate. Then phosphoglycerate kinase, it catalyzes the reaction between 1,3-bis phosphoglycerate plus ADP to form 3-phosphoglycerate plus ATP. And it is a reversible kinase reaction. Pyruvate kinase also doesn't catalyze a reversible reaction. So the answer here is option C, that is phosphoglycerate kinase. All the enzymes are taken from glycolysis. So next one. At high glucose levels, fatty acid catabolism is inhibited by the inhibition of. So it is by carnitine acyl transferase by malonyl CoA. So carnitine acyl transferase, it is involved in fatty acid oxidation, and it is inhibited by malonyl CoA. So at high glucose levels, fatty acid catabolism is inhibited by malonyl CoA by inhibiting carnitine acyl transferase. This inhibition, it causes decrease in the transport of long chain fatty acids into muscles and heart mitochondria, decreasing fatty acid oxidation in heart and in muscle and heart mitochondria. So answer is option A. Then next one, DNA glycosylases are involved in dash repair. So DNA glycosylases, they are involved in base excision repair. Answer is option C. Dash has 5' to 3' exonuclease activity. So it is DNA polymerase 1 that is having 5' to 3' exonuclease activity. Clino fragment, it possesses 5' to 3' polymerase and 3' to 5' exonuclease activity. But it doesn't have 5' to 3' exonuclease activity. It is possessed by DNA polymerase 1. The next one, find out the odd one. GTPase activating protein, RAS pathway, guanine nucleotide releasing protein, MAP kinases. So first three, that is GTPase activating protein, RAS pathway and guanine nucleotide releasing proteins, they are involved in the conversion of GTP to GDP or GDP to GTP. Whereas the MAP kinases are specific to amino acids serine and threonine. So the odd one is option D, that is MAP kinases. The next one, how many different RNA polymerases are found in E. coli? Only one. Then find out the odd one, Scrappy, CDJ, ataxia and Kuru. So the Scrappy, CDJ and Kuru, they are caused by prions, whereas ataxia is not caused by prion. So answer is option C. Find the incorrect match. Shine, Delgarno and Cossack. So it is a ribosome binding sequence in prokaryote, and Cossack is ribosome binding sequence in eukaryotes. Option B, RBS and Cossack. Cossack is an RBS, so that is also correct. Promoter and Tata box, that is also correct. Whereas promoter and Shine-Dalgarno. Shine-Dalgarno is not a promoter. So option D is the incorrect match. The next one. Which of the following statement about promoter in PSPAC is wrong? It is from Phage SPO1. That is correct. It can be induced by IPTG. That is also correct. It can be repressed by lac repressor. That is also correct. It is used for regulated expression of foreign genes in Pseudomonas putida. That is not correct. So answer is option D. The next one. Find the odd one out. Topo cloning technology. So it is a technology of cloning which does not require DNA ligase. BP clonase, LR clonase gateway technology. So what is gateway technology? It enables for researchers to transfer DNA fragments between plasmids, but or I can say between various cloning vectors, because it has got gateway ATT sites as well as two enzyme mixes, that is LR clonase and BP clonase. So find out the odd one out is topocloning technology, whereas BP clonase, LR clonase and gateway technology all come under the same heading, gateway cloning. Next one. What is the biosafety level practices recommended for SARS-CoV-19? So it is biosafety level 3. Answer is option C. Then recombinant hepatitis B vaccine is a preparation of. So hepatitis B vaccine, it is composed of hepatitis B surface antigens absorbed onto aluminium adjuvant. So answer is option D, that is HBsAg. Then HAT medium is composed of. So it is composed of hypoxanthine, aminopterin, thymidine. So that is found in option D. Answer is option D. Then HGPRT is necessary for. It is necessary for salvage synthesis of nucleic acids. Answer is option A. These are all direct questions. That is why I am just giving out the answer without much explanation. Then ELISA is enzyme linked immunosorbent assay. So answer is option C. Then BCG vaccine contains. It contains live bacteria that has been weakened and it is attenuated vaccine. Answer is option B. Then TATA is. So TATA is tumor associated transplantation antigen. Answer is option B. Which of the following classes of antibody significantly crosses the human placenta? It is IgG. Answer is option A. This is also a usual question for many entrance examination. Not only for SET exams, they have asked even for CSIR, DBT, other university exams, other university PhD exams. For all that, they have asked this question. Then, which among the following statement correctly explains the autonomy argument against human cloning? So answer is option D. Each individual has the right to develop without interference. Then next one. Find out the odd one among the following thermostable polymerases. So all these are thermostable polymerases. You have to choose the odd one. So out of these, Taq polymerase, deep vent polymerase, Tilly polymerase, Pau polymerase. The odd one is Taq polymerase. Why? Because fidelity of the other three, that is deep vent polymerase, Tilly polymerase, Pau polymerase, is higher than Taq polymerase. So answer is option A. Then next one. Which statement among the following is wrong about PUC plasmid? It works by alpha complementation. That's correct. Recombinants can be identified by blue-white screening. Yes, that is also correct. It contains beta-lactamase gene. That is also correct. It is based on PSC101 replicon. No. So option D is the answer. Then next one. Select the correct statement. AFLP is a codominant marker. It is a dominant marker, not a codominant marker. So that statement is not correct. RFLP is PCR based marker. RFLP is a non-PCR based marker. So that is not correct. SNP are codominant markers. That is correct. Then RAPD uses 20 mer primers. It is not 20 mer, it is 10 primers RAPD uses. So the answer is option C. Then next one. Which of the following next generation sequencing protocol is not based on sequencing by synthesis? It is AB solid. Answer is option B. Then next one. Which among the following is not true about oligonucleotide adapters for restriction and modification? They are formed by partially complementary oligos. They do not have preformed sticky ends. That is not true. So answer is option B. Which among the following is not true? They have asked regarding adapters. So they do not have preformed sticky ends. That is not true. Then next one. Statement 1. Metaplasia is the conversion of one tissue specific stem cell to another. Then in transdifferentiation, one differentiated cell type become another. So they have asked if both the statements are correct. And the answer is option A, that is both 1 and 2 are correct. The next one. SARS-CoV-2 is a dash enveloped virus. So it is a single stranded RNA virus. Everybody knows that. It was there in papers. So SARS-CoV-2 is a single stranded RNA virus. Then chemolithotrophs gain energy from. So they gain energy from reduced inorganic compounds. Answer is option C. Then next one. Penicillin kills susceptible bacteria by dash transpeptidase. So by specifically inhibiting the transpeptidase. Answer is option C. Then lysozyme acts by hydrolyzing the bond between NAG and NAM, N-acetylglucosamine and N-acetylmuramic acid. So answer is option A. Then daily production of which of the dash is greater than that of any other immunoglobulin class. So it is secretory IgA. Answer is option B. Then paratope is a part of. So paratope, it is the part of an antibody which will recognize the antigen and bind to the antigen. So it is a part of the antibody. Answer is option A. Then alteplase is a recombinant form of dash activator. It is a tissue plasminogen activator. Answer is option C. The technique which is not used specifically for avoiding spurious PCR amplicons. So hot start PCR can be used for avoiding these spurious PCR amplicons. Nested PCR can also be used. Then inverse PCR. Inverse PCR was designed for amplifying anonymous flanking genomic DNA regions. So it is not for avoiding any spurious PCR amplicons. So answer is option C. Then next one. The protocol which is not used for screening expression libraries, hybrid arrest translation, that cannot be used. Immunological screening, southwestern screening, northwestern screening can be used for screening expression libraries. So answer is option A. Then next one. Callus is an undifferentiated tissue with differing ploidy. Callus differentiation leads to somaclonal variation. So both 1 and 2 are correct. And 1 is the correct explanation of 2. That is also correct. Because callus is an undifferentiated tissue with differing ploidy and it can lead to somaclonal variation. So both 1 and 2 are correct and 1 is the correct explanation of 2. The next one. Chloroplast transformation is advantageous because biological containment of transgenes can be ensured. Yes. Dosage effect for transgene expression is obtained. Position effects can be minimized. All of the above. So all of the above is the answer. Then dash is an autosomal dominant disorder. Huntington's disease, Engelmann's disease, Duncan's muscular dystrophy and cystic fibrosis. So out of these autosomal dominant disorder is Huntington's disease. Answer is option A. Then next one. Which among the following intellectual properties come into existence as soon as invented without any further procedures? So as soon as invented and without any further procedures, it is your copyright. So answer is option A. Then next one. The geometric mean of 2 and 8 is. So we know the formula for geometric mean. It is square root of n x1 into x2. So n is 2. Only 2 digits are given. So square root of x1 is 2 and x2 is 8 and number of terms is 2. So square root of 2 into 8, that is 16, is equal to 4. Answer is option D. Then next one. The distance between Cartesian points 2, 3 and 5, 7 is. So distance between, these are just your basic maths questions. So distance between 2 points is given by square root of x square minus x1 the whole square plus y2 minus y1 the whole square. So here x1 and y1, 2 and 3 and 5 and 7 are x2 and y2. Substitute in the formula, you will get answer 5. Answer is option A. Then next one. Mesosomes differ from intrusion bodies in. So they differ from intrusion bodies in their structure, function, location. Answer is all of the above. Then next one. Bacterial spores are rich in calcium dipicolinate. Answer is option A. According to Indian patent law, the term of a patent is for. So it is for 20 years from the application of the patent. Answer is B. Then next one. Which among the following statement is wrong? Genetically modified organisms are patentable in India. India follows sui generis system of plant variety protection. Plants are patentable in India. India is not a member of UPOV, that is International Union for the Protection of New Varieties of Plants. So the answer here is option C. Plants are patentable in India. That is the wrong statement. The next one. The form used for filling complete or provisional specifications of patent in India. It is form 2. Answer is option B. Select the correct alternative. Trademark can be used for infinite period. That itself is correct. Answer is option A. Then Budapest Treaty is concerned with. So it is concerned with the deposit of microorganisms. Answer is option C. Then which among the following change has happened to Indian patent law after TRIPS? TRIPS is trade related aspects of intellectual property rights. Change from process to product patent. That itself is the answer. Answer is option A. The term petty patent is used for dash patent. So it is used for utility patent. Answer is option D. Then the major carriers of salmonellosis. It is meat and eggs. There is some printing mistake in the option, but the answer is option A, meat and egg. Then the organism responsible for bloody bread. So it is Serratia marcescens. Answer is option B. Next one. Find out the roots of the quadratic equation x square plus 5x plus 6. So we will factorize by splitting the middle term. So 5x can be split into 3x plus 2x. That will give you 5x. And 3 into 2, it will give you 6. So it can be written as x square plus 3x plus 2x plus 6. Now take out the common factor from the first two terms. This is all your basic maths in your school. So x square plus 3x is equal to x into x plus 3. So take the common factor out from the third and fourth term. So that is 2x plus 6. This is the third and fourth term. 2x plus 6. This is the first and the second term. So that will give you 2 into x plus 3. Then you can write x into x plus 3 plus 2 into x plus 3 for this particular equation. Now x plus 3 is common, so you take it out, you will get x plus 3 and x plus 2, which are the factors for x square plus 5x plus 6. And to find out the roots, just do it like this. X plus 3 is equal to 0, so x will be minus 3, and x plus 2 is equal to 0, so x will be minus 2. So the answer is A, that is minus 2 minus 3. Answer is option A. Next one. Find the sum of 100 terms of the arithmetic progression 2, 4, 6. So sum of the first n terms of an arithmetic progression. It is given by Sn is equal to n by 2 2a plus n minus 1d. So a is the first term, so that is 2. D is the common difference. So second term minus first term. So 4 minus 2 is equal to 2. N is the number of terms. So that is 100. Now substitute in the formula, you will get 10100. So answer is option C. Then next one. Aerobic method used for wastewater treatment. So they have asked for aerobic method. So answer is option A, that is trickling filter. Industrial alcohol will be produced by using starter culture. So the starter culture used for industrial alcohol production is bottom yeast. Answer is D. The enzyme used for fruit juice clarification, it is pectinase. Answer is C. The delayed ripening of tomato was created by silencing. So answer is option D, silenced. Then the organism under GRAS status. GRAS is generally recognized as safe. So the organism under GRAS status will be lactobacillus. If you know the full form of GRAS itself, you can come to know that lactobacillus is the answer. Then select the correct statement. Genes without a known function are not patentable. That is correct. So answer A itself is correct. All the others are not correct. Okay. Design Office of India is situated in. So it is situated in Delhi. Calcutta. Answer is option D. Then next one. The publication of the patent is done in India after dash months of application. So answer is 18. Answer is option B. Then next one. Find out the odd one. Rax ML, Mr. Base, Pop and S Diva. So Rax ML, that is randomized accelerated maximum likelihood, Mr. Base and Pop. Pop is phylogenetic analysis using parsimony. All these three, they are involved in phylogenetic analysis. S Diva, that is statistical dispersal variance analysis. It is used for inferring biogeographic histories. So the odd one out here is option D. The next one. Find the wrong statement. DDBJ is a nucleic acid sequence bank. Yeah, that is correct. DDBJ of Japan. Then Sequin is a standalone platform for sequence submission. That is also correct. BankLT is a standalone platform. BankLT is not a standalone platform. Only Sequin is the standalone platform. So the wrong statement is option C. Then next one. The tree building algorithm based on distance matrix. So the one based on distance matrix is UPGMA. Answer is option A. Then next one. How many reading frames are found in double-stranded DNA? So DNA, it is interpreted in groups of 3 nucleotides, or I can say codons. And DNA strand has 3 distinct reading frames. Now in the question it is asked for double-stranded DNA, so it must have 6. How? 3 on the positive strand and 3 on the negative strand. Positive strand matlab the parallel strand, and the negative strand means anti-parallel strand. So the answer here is option D, that is 6. The next one. Chinese red rice is. So Chinese red rice is Angkak. Answer is option C. Then, the byproduct of dairy industry used as carbon source in fermentation media is naturally whey. They have asked for dairy industry byproduct. Molasses, dextrine, corn steep doesn't come under dairy industry. So it is whey. Then an example of sedimentary type of nutrient cycle. So it is phosphorus cycle. Answer is option C. The algae which produce hydrogen gas. It is Chlamydomonas. Answer is C. Then radiant energy is converted to potential energy. Radiant energy is energy from the sun converted to potential energy by producers. So answer is option B. Biodiesel is obtained from Jatropha curcus. This is also a repeated question for many entrance examination. Answer is option C. Gas having highest greenhouse effect. It is N2O. Answer is option D. Which among the following is an organophosphate pesticide? So it is parathion. Answer is option B. Which following combinations is acid rain? So it is H2SO4 and HNO3. Here also there is some printing mistake, but answer is option C, H2SO4 and HNO3. A scientist wishes to produce seedless watermelons for commercial cultivation by tissue culture. What might be his technique of choice? So seedless watermelons, they are triploid. So endosperm culture is sufficient for the production of seedless watermelons by tissue culture. So answer is option C. The next one. A scientist wishes to rescue a rare mutant ornamental plant from viral attack. His technique of choice will be. So it is. So he want to rescue it from viral attack. So naturally, meristem tip culture will be the method of choice. Now this question also has been asked in different ways, but the answer is the same. It is meristem tip culture. But the question is been asked in different ways for various entrance examination I have observed. Zolgensma, a drug used for treating spinal muscular atrophy, is made using. So patients with the spinal muscular atrophy, they will have a defect in the gene known as SMN1. And it is required for normal functioning of nerves that control the various muscle movements. And Zolgensma, it is made up of a SMN1 gene which is working, and it is placed in a vector, and the vector is adeno-associated virus 9 or AAV9. And the vector, what it does, it will deliver the working or the good SMNV1 gene to motor neuron cells throughout the body. So they have asked, Zolgensma drug used for treating spinal muscular atrophy is made using adeno-associated virus. Answer is D. The next one. The addition of microorganisms which favor health benefits. It is probiotics. Answer is option A. Miso is fermented product of rice. Answer is option A. First amino acid commercially produced by fermentation. It is L-lysine. Answer is B. The next one. Role of microorganism in fermentation was discovered by Louis Pasteur. Answer is A. So coming to the next question. Clostridium perfringens poisoning is associated with. So it is perfringens they have asked. So it is meat product. Answer is option B. A die is rolled 3 times. Find the chance. The first roll is an ace. The second roll is a deuce and the third roll is a tray. So the first roll is an ace is 1 by 6. Second roll is a deuce, that is also 1 by 6. And third roll is a tray, that is also 1 by 6. So all three coming together. So 1 by 6 into 1 by 6 into 1 by 6. So 1 by 216. Answer is option C. A deck of cards is shuffled and 3 cards are dealt. Find the chance that the first card is a king. So answer is 4 out of 51. Answer is option D. The next one. pcDNA is a plasmid with SV40 origin of replication. Correct. pcDNA can replicate in murine cell lines. That is also correct. Okay, both statements are true, but one is the correct explanation of two. Both statements are true. One is not the correct explanation of two. One cannot be the correct explanation for 2. So answer is option B. So that's it. Thank you for watching my channel. You can also refer my other videos on SET Biotechnology exam conducted by Kerala SET of Kerala SET exams biotechnology. You can refer my channel or previous year question papers. You will get it from 2019 onwards. I have answered it with explanation. So thank you for watching. Please do like, share and subscribe. Thank you.